and movement lesson, chapter 9. Okay, so here in this video we are going to see the structure and its function of the muscle fibers. So here what we are going to see is we are going to see the structure of myofibrils, which appear as a dark and light bands when it is seen under the microscope. Okay, so the dark band is named as A band and isotrophic bands, and the light band is termed as I bands, isotrophic bands. So these are perfectly aligned with one another. Okay, so one after another it is aligned perfectly, and when we see this arrangement one after another, it look like a striated appearance. Okay, that means it look like a layers. So each of this dark band, inside that dark band we have a lighter region in its middle. So in a dark band, that is the A band, a middle region is present, that is lighter. Okay, that is termed as H zone. H zone means Hellas means clear zone. So that look like a clear zone when we see it in depth. Okay, so now clearly I am saying A band has a lighter region in the middle, that is called as H zone, which is considered as a clear zone. Okay, now in this H zone, if you bisect it vertically, there is one dark line, so that is called as M line. Okay, dark line is known as M line. So in A band there is a lighter zone called H zone, and inside that there is one dark line, that is called as M line. So this is the structure of the A band. Now coming to the light I band, likewise in the A band, here in the light I band, again the opposite is the darker for the light. So here we have a darker midline area, that is called as Z disc. So Z disc, that is actually found between the two I bands. Okay, so Zwischenzeb. Okay, Zwischenzeb. So that is the pronunciation of the Z when you expand it. So this is the structure of the A band and the I band. Okay, so how will be the arrangement? I band, A band, I band, like that it will be a arrangement. But in I band, the midline, no, so if you can see the midline between the I band. Likewise in the A band, the midline M is also found between the in center of the A band. Okay, this you people have to remember. Now coming to the myofibrils present in the muscle. So in myofibril contain a contractile element that is termed as the sarcomere. Okay, the contractile element, the sarcomere, which is considered as the functioning unit of the skeletal muscle. So in when the contraction has to be performed, who is performing? The sarcomere is performing. So which is the functional unit of the skeletal muscle. And if you see the sacromere, the myofibril, this is present, the sacromere is present in the region between the two Z disc. Now remember what is Z disc? Z disc என்றது என்ன அப்படினு பார்த்தா, where it is found? It is found between the I band. இல்லையா? So this one is there only the sarcomere is found. So it contains an A band with the half I band. Okay, so now if you see, A band is present, then half I band is present at the end of the muscle. So inside this I band only Z disc is there. So inside that only we have the sarcomere. Now if you take the sarcomere is made up of two filaments. One is thick filament and another one is thin filament. So the thick filament which is present to the whole length of the A band. Okay, so actually this sarcomere is present in the Z disc, but the extension of the thick filament of the sarcomere will extend towards the A band. Okay, and the thin filaments will extend across the I band. I hope you understood. Okay, next, and the partly into the A band. Okay, this will give us the complete structure of the muscle fibers. Okay, so now coming to the sarcolemma. What is sarcolemma? The plasma membrane of the muscle fiber. Okay, so that is called as sarcolemma. So it forms a T tubules. So it forms a transverse. T means transverse tubules like structure, and that will penetrate. That means that will get inside the junctions between the A and the I band. So this you can see clearly in the given picture. Figure 9.1. Okay, see that here we have first one, we have a dark A band, then we have a light I band. Okay, so lighter and darker bands. So if we see this in depth, we have a tubular structures in center. So all the tubular structures only we call it as the myofibrils. So when you take this myofibrils out, you can see this expansion, that is the dark A band and light I band. Okay, and see that in the dark A band in center we have the lighter band. Okay, and likewise in the I band we have a darker band. That is what we call it as the Z disc. When you expand this Z disc, inside that we have the sarcomere. Okay, that is what given in the third picture, and in that see that the sarcomere is extending, the thick filament is extending towards the this one IA band, and the light one is penetrating inside the I band. Okay, so that is what they have given in this picture. So this is how the structure look like. Now coming to the structure of the contractile protein. I have said two proteins are present. One is actin and myosin. This two only we term it as the myosin in the myofilaments. Okay, so these two are present, actin and myosin are present in the myofilaments. So the thick filament, I said no, two filaments are there, thick filament and thin filament. Thick filament is composed of myosin. Okay, and it contains a head-like structure. That structure will be protrude out. Okay, so in each of this which are bundled together, the heads produce at opposite ends of the filament. Each of the opposite end there is a head-like structure. So each of the myosin molecule actually is made up of this structure, one single structure, that is called as monomer, one single structure that we termed as meromycin. So the meromycin have two regions, and globular head and a tail-like structure, arm and tail-like structure, that is a short arm and a tail. So when you take this meromycin, you can see there are two regions. One is the globular head and another one which is look like a tail. Okay, so now coming to the short arm. Okay, so that is made up of heavy meromycin, and the tail part is made up of light meromycin. So that is what we termed here at it as the HMM and LMM. Okay, so the head now the head tail part. Okay, so the head part is having the site for actin binding and ATP binding, and it produces the ATP enzyme also, which is needed for the splitting of the ATP, which is generated, which is needed for the contraction of the muscle. So now you say where the ATP is splitted up and where the ATP and the actin binding site is there. It is in the heavy head part of the meromycin. Okay, so the thin filament, now it is composed of actin molecules. Okay, that will intertwined with each other. Intertwined. So this coming to the actin, actually myosin, we have seen that it is made up of actually it is made up of this binding sites, head, tail and short arm, like that we have seen, and it is a monomer. But in actin it is a polypeptide subunit, many is there. So it is looking like a bead like structures. Okay, so then it look like a protein, globular protein. Global means round, right? So it is termed as globular actin or G actin. Okay, and it rolls upon, right? The intertwined ஆயிருக்கிறதுனால and the filamentous forms அது வந்து form பண்ணுது. So it look like a filament by itself. So we call it as the F actin. Okay, if in this depth, if we see, it is helically wound with each other, so it is made up of a polymer of monomeric reactants, right? So it is, what is the site here? It is a binding site for the myosin. Actually, meromyosin, which is the binding site, that is the thick filament which is made up of protein myosin, that is having a site for the ATP binding site. So which is what we said, it has the head and the short arm. And if you compare it now here, the binding site present in the actin is for the myosin, okay, and the thin filaments having some other protein also, which is termed as tropomyosin, troponin, all this, okay, regulatory protein. So regulatory means it will regulate the contraction of the muscles. Along with the actin and the myosin, this protein also involved in the contraction of muscles. So in figure 9.2 it is clearly given. Okay. It is clearly given how the thick and the thin filaments are present.